Today we will discuss about the effect of chronic nerve injury and how the immune cells react to this chronic nerve injury in terms of this metabolic support that is provided by astrocyte, and this microgalia is the immune cells that helps in responding to this chronic nerve injury. So we will start with this microgalia. So in this microgalia, what happens is that the glucose intake will increase whenever there is chronic nerve injury, and because of this it will lead to this anaerobic glycosylations, and that will cause the upregulations of these genes, that is PEP, SUC, CITR, alpha Kg, and thereafter what will happens in order to make up for the Ca2 plus that is required at the endoplasmic reticulum. There is a release of this lactate that occurs through these receptors, that is through this uh, cell membrane pore, that is MCT 1 by 4, and the release of this lactate will cause the Ca2 plus to enter into the cells and goes into this endoplasmic reticulum. And because of the increase in the amount of Ca2 plus, it will cause the upregulations of these pro-inflammatory genes such as HIF 1 alpha, mTOR, NF kappa beta, interleukin gamma, and this will leads to the pro-inflammatory response or inflammations because of this chronic nerve injury. And how the metabolic support is provided, now we will understand the metabolic support that is provided by astrocytes. So astrocytes have the ability to intake more glucose and convert it into the pyruvate. So whenever there is a chronic nerve injury, what will happen is that astrocytes will increase. There is an increase in the glutamate intake by the astrocytes that will cause increase in the glucose intake, and that can be converted into the pyruvate through this PFKB B2 by 2 or 3. This is not 1 by 4, this is MCT1 or MCT4, and this is also PFKB2 or PFKB3. And this pyruvate then will cause this glucose to go under anaerobic glycosylations that will lead to this high lactate formations. At the same time, it will also upregulates the key enzymes that helps in this glycosylations. That is, and these key enzymes will then help in this anaerobic glycosylations to form lactate, and that can be released from MCT1 or MCT4 pores of the astrocyte cell membranes. And these lactates can then be intake by the neuron through this MCT2 uh, pores or this cell membrane uh, proteins or receptors that are present on the neuron. And this lactate intake is necessary in the case of neuron to produce the ATP, which is the energy, which is in general terms the energy that is required by the cells to perform its regular functions. At the same time, the genes that are involved in oxophosphorilations are also present or also upregulated in this neuron that will help in the TCA cycles. So in both, the ATPs are produced through these upregulations of these oxphos genes. At the same time, the lactate intake by MC2 receptors that is produced by both microgalia and astrocytes, and the glucose intake by these GLUT receptors. So all these things will cause these pyruvate reactions to form, and thereafter goes into the TCA cycles, and that will lead to the formation of this ATP that can help in the performing the regular functions that is required by the cells or the neurons. So in brief, this microgalia is the immune cells that respond to the chronic nerve injury, and astrocytes is the astrocyte cells are the cells that provides metabolic support to the neuron to perform its regular functions. So this is how the microgalia, astrocytes, and neurons are related, and how they react whenever there is a chronic nerve injury. If you need to understand more about this chronic nerve injury in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.